Okay, let's dive right into this explainer on the chromosomal basis of inheritance. We're going to be exploring exactly how these microscopic rulebooks dictate the traits of literally all living things. If you've ever wondered how the abstract idea of a gene physically translates into the color of your eyes, the shape of a leaf, or even the underlying causes of certain human diseases, you are absolutely in the right place. But hey, before we get into the really heavy medical applications and intricate cellular mechanics, we gotta start with a very relatable moment of pure frustration. These are the actual words of embryologist Thomas Hunt Morgan in his 1907 fly room over at Columbia University. He spent months just staring through a microscope at thousands of tiny fruit flies, trying to find just one single natural variation to study. It's a pretty great reminder that behind every massive scientific breakthrough is usually a researcher who was, at one point, absolutely exhausted. So, to make this dense information super digestible for our 10-minute lecture today, we're going to break things down into 5 key sections. 1. The fly room discoveries. 2. The sex chromosome factor. 3. Linked genes and recombination. 4. Chromosome errors and disorders. And 5. Exceptions to the rules. We're essentially tracking how these rules were written, their specific nuances, and then what goes completely wrong when they break down.
All right, part 1. The fly room discoveries, building the rulebook. So Morgan's persistence actually did pay off. After 2 whole years of seemingly wasted effort, he finally spotted a single male fruit fly that completely broke the visual norm. And this tiny insect was the key to proving that chromosomes are indeed the physical location of Mendel's heritable factors.
Let's just set up our baseline vocabulary real quick. In genetics, the trait we see most commonly in natural populations, like red eyes in a fruit fly, we call that the wild type. Basically, that is the rule. The alternative trait, like that weird white-eyed anomaly Morgan found, that's the mutant phenotype. And that is our very first rule breaker, representing an allele that's assumed to have originated as a mutation. So Morgan takes this anomalous white-eyed male and breeds it with a regular red-eyed female. Eventually, in the second generation of offspring, the white-eyed trait reappeared, but with a massive catch. It only showed up in the male flies. By breeding them, Morgan figured out that eye color was directly tied to the biological sex of the fly. Since males have one X and one Y chromosome, a single mutant allele on their sole X chromosome gave them white eyes. This was a literal game changer. It proved for the absolute first time that specific genes live on specific chromosomes. Moving right along to part 2, the sex chromosome factor. And this is about way more than just sex. Morgan's flies showed us that the X and Y chromosomes carry whole genetic rulebooks of their own. Because as it turns out, nature has written a bunch of different rulebooks for determining sex across different species. We humans and other mammals use the XY system, where the presence of the SRY gene on the Y chromosome pulls the trigger on male development. But look at how wild the diversity is here. Grasshoppers use an X0 system, where males just have one sex chromosome. Birds use a ZW system, where the egg, not the sperm, dictates the sex. And bees, they don't use sex chromosomes at all. The males just develop from unfertilized eggs.
But jumping back to mammals for a second, female cells have this really unique challenge. They inherit 2 X chromosomes, while males only have 1. So to prevent a double, highly toxic dose of X-linked proteins, female mammals undergo a process called X inactivation. During early embryonic development, one X chromosome in each cell is just randomly silenced by RNA and methyl groups, and it condenses into this dense little object called a bar body. This creates a literal mosaic of cells. Half your cells express your mother's X chromosome, half express your father's. Fun fact, this is exactly why female tortoiseshell cats have those awesome patchy orange and black coats.
All right, part 3, linked genes and recombination. Getting into the nuances. So, we've established that chromosomes carry genes. Humans have hundreds, even thousands of genes on a single chromosome. But what happens when 2 completely different genes live right next to each other on the exact same chromosome? Morgan actually wondered this too. He looked at 2 traits: body color, gray versus black, and wing size, normal versus tiny, vestigial wings. When he bred heterozygous flies, he fully expected a perfectly even 1:1:1:1 ratio if the genes sorted independently. But the actual results? They told a very different story. The vast majority of the offspring looked exactly like the parents, either gray with normal wings or black with vestigial wings. The parental traits largely stuck together, meaning these 2 distinct genes were physically tethered. They were linked on the same chromosome.
But of course, there were some rule breakers. A small chunk of the offspring had totally non-parental combinations, like a gray body with vestigial wings. So how did linked genes separate? Well, this led to the discovery of crossing over. During meiosis, when homologous chromosomes pair up, their chromatides occasionally trade places, literally swapping corresponding segments of DNA. It basically shuffles the genetic deck, breaking the physical connection between linked alleles and generating some absolutely vital genetic variation.
Since these mixed-up recombinant offspring made up precisely 17% of the total, Morgan student Alfred Sturtevant had a massive realization. He hypothesized that the further apart two genes are on a chromosome, the higher the probability that a crossover event is going to happen between them. Scientists realized they could use these recombination frequencies, like this 17%, as literal map units on a highway to measure the physical distance between genes and actually map the entire chromosome.
Which brings us to part 4, chromosome errors and disorders, or what happens when the rules break down. Now, why does all this microscopic mapping matter to us? Well, because understanding the precise order and behavior of this rulebook is absolutely crucial for understanding the severe human health impacts when those rules break down during cell division.
You see, our cellular machinery is usually incredibly precise, right? But occasionally there's a mechanical mishap called non-disjunction. This is when a pair of homologous chromosomes simply fails to move apart properly during meiosis. One gamete gets 2 copies of a chromosome and the other gets 0. When these flawed gametes are fertilized, it leads to aneuploidy, which is an abnormal number of chromosomes in the resulting zygote. And this is the underlying cause of conditions like Down syndrome or trisomy 21, meaning there are 3 copies of the 21st chromosome. Aside from that uneven sorting, the physical structure of the chromosomes themselves can also get damaged by radiation, chemical disturbances, or errors during crossing over. And this happens in 4 primary ways. A deletion completely removes a chromosomal segment. A duplication repeats a segment. An inversion reverses a segment backward inside the chromosome. And a translocation takes a piece of one chromosome and drops it onto a completely different, non-homologous chromosome. Each of these brings a really unique and often severe set of developmental consequences. To put that into perspective, let's look at the absolute devastation a simple microscopic swap can cause. If a specific large fragment of chromosome 22 accidentally swaps places with the very tip of chromosome 9 during the mitosis of white blood cell precursors, it creates an abnormally short chromosome 22. We call this the Philadelphia chromosome. This structural error creates a brand new fused gene that leads to uncontrolled cell cycle progression. Basically, this tiny translocation is the direct cause of chronic myelogenous leukemia, a dangerous form of cancer. Finally, Part 5, Exceptions to the Rules. We're looking at imprinting and extranuclear DNA. As we approach the end of our explainer here, having covered the rules and what happens when they break, we have to confront the ultimate rule breakers. These are naturally occurring genetic systems that completely ignore standard Mendelian laws.
Now, under standard Mendelian rules, it really doesn't matter if you get a dominant allele from your mom or your dad, the result is exactly the same. But in genomic imprinting, the old rules are completely erased. During sperm or egg formation, chemical markers literally silence certain alleles based entirely on which parent they came from. For example, in mice, the IGF-2 gene is required for normal growth, but the mother's copy is always silenced. So if a mouse gets a mutated, dwarf-causing allele from its father, it's going to be born a dwarf. But, and this is crazy, if it inherits that exact same mutant allele from its mother, it grows completely normal because the mother's defective copy is naturally ignored.
And get ready for this, because you actually have DNA living completely outside of your cell's nucleus. You have small, circular DNA molecules just hanging out in your mitochondria, the powerhouses of your cells. Because the cytoplasm of a fertilized zygote comes almost entirely from the mother's egg, these mitochondrial genes are inherited strictly from your mother. Defects in this extranuclear DNA totally bypass standard genetics and lead to really unique maternal inheritance patterns that primarily impact your energy-hungry systems, causing severe muscle deterioration and nervous system disorders. And that brings us to a really fascinating frontier. Researchers have found that as we age, new mutations gradually accumulate in this exclusively maternal mitochondrial DNA. This decay limits cellular energy production over a lifetime and is believed to actively contribute to major age-related conditions like heart disease and Alzheimer's. So, as mutations in our mitochondrial power plants dictate our decline, it really leaves us wondering,
What other genetic mysteries are waiting to be unraveled inside our own cells? And is aging itself just another genetic rulebook we simply haven't figured out how to rewrite yet?